Imagine a future where the power of supercomputing isn't housed in sprawling data centers, but instead operates silently within a liquid, harnessing the very essence of life itself.
That's right.
Okay, let's unpack this because it might sound like science fiction, but it's rapidly becoming reality.
It really is.
Today, we're taking a deep dive into the fascinating world of DNA-based supercomputing. Our focus. The recent groundbreaking work from Dr. Fei Wang's team at Shanghai Jiaotong University and their creation of DNA-based programmable gate arrays, or DPGAs.
DPGAs.
Think of it as a revolutionary new approach to processing information.
That's right. What Dr. Wang's team has achieved, and it's detailed in their Nature publication alongside other, cutting-edge research, it isn't just incremental progress.
Right.
It's really a fundamental shift in how we think about computation.
Exactly. So our mission in this deep dive is to make sense of this exciting development for you. We want to understand the core ideas, what makes it tick.
The principles behind it.
Yeah, and explore the truly mind-boggling potential it unlocks, and consider the significant implications it holds for the future of technology in our lives.
It's a big topic.
It is. Consider this your shortcut to getting a solid understanding of this complex but potentially game-changing field. We've sifted through the research papers that key Nature publication included, expert reports, a wealth of analysis.
A lot of material.
To bring you the most important nuggets, all while keeping it engaging and hopefully a bit surprising.
Well, what's truly revolutionary here is the foundational concept. Using DNA, the very blueprint of life, to actually perform calculations.
Right.
This places it squarely within the burgeoning field of molecular computing, where the aim is really to emulate the incredibly efficient information processing we see happening constantly within living cells.
And it strikes me that this isn't something one type of scientist can just figure out on their own. It must require a real mix of expertise.
Absolutely. It's a beautiful example of interdisciplinary science. You need deep knowledge from chemistry, biology, bioengineering, even material science.
Wow.
Dr. Wang's work perfectly illustrates this synergy where these seemingly distinct fields converge to produce something entirely new and powerful. If you think about the bigger picture, many of the most significant technological leaps happen right there at these intersections of different scientific domains.
So, at the heart of this is DNA. We all know it carries our genetic information, but how do you go from that to, performing calculations?
Okay, so the fundamental principle is that DNA acts as an incredibly dense information storage medium. Think of the 4 nucleotide bases, A, T, C, and G.
Right, the letters.
As being like the zeros and ones in a computer's binary code. Each of these bases can essentially represent a bit of information. It's almost like life has already solved the problem of storing vast amounts of data in a tiny, tiny space.
Okay, so DNA holds the data, but how do you actually manipulate it to do something, to perform a calculation?
Right, so computation happens by carefully controlling the interactions between these DNA molecules in a lab setting.
In a test tube, basically.
Pretty much, yeah.
Yeah.
Researchers use a range of techniques, like PCR, which you probably know is like a molecular photocopier.
Right, yeah.
To make many copies of specific DNA sequences. Then there's gel electrophoresis to separate DNA fragments based on size, and DNA sequencing to, read the precise order of those A, T, C, and G bases.
Okay.
And crucially, they use specific molecular operations to perform the logical steps.
Can you give us some examples of those molecular operations?
Certainly. So imagine one DNA strand bumping into a double-stranded DNA molecule and essentially kicking one of the existing strands out. That's DNA strand displacement.
Okay.
Then you have hybridization, where 2 complementary DNA strands recognize each other and bind together, like 2 puzzle pieces fitting perfectly.
Right.
And then there are more sophisticated techniques, some with catchy names like toe-hold-mediated strand displacement or polymerase-based strand displacement and toe-hold exchange.
Wow, okay.
These are the fundamental tools for building logic gate, your basic AND, OR, and NOT gates.
Like in a normal computer chip.
Exactly. And even for amplifying signals within what are known as chemical reaction networks, or CRNs. Think of CRNs as sort of miniature chemical circuits.
So we're taking these fundamental biological interactions, the same kind of things happening inside our cells every second, and we're engineering them to act like the tiny switches in our computers.
Precisely.
That's a mind-blowing thought. It's like we're learning to speak the language of life to get it to do our bidding.
That's a good way to put it. And the key insight here is to draw a direct analogy with traditional silicon-based computing. You can think of DNA molecules as acting like the wires or the instructions, maybe even the electrons in these biological circuits.
Hmm.
And these DPGAs we're focusing on are directly inspired by FPGAs, field programmable gate arrays.
Okay, FPGAs. Remind us what those are.
Right, so if you're not familiar, think of FPGAs as essentially blank silicon chips that engineers can configure after they're made to perform all sorts of different tasks. Very flexible.
Oh, okay.
DPGAs aim to bring that same reconfigurability, that same flexibility, down to the molecular level.
Now, here's where it gets really interesting. One of the big touted advantages of DNA systems is this concept of massive parallelism. What does that actually look like in a test tube?
Okay, imagine a traditional computer tackling a problem step by step, sequentially.
Yeah.
Now, picture billions, even trillions of DNA molecules floating in a solution, all interacting and processing information simultaneously.
Wow.
That's the power of massive parallelism. Because DNA molecules are so incredibly small, you can have an astronomical number of them working on different parts of a problem at the same time, all within a tiny volume.
Hmm.
This is particularly beneficial for tasks that can be broken down into lots of independent calculations. Think of it like having an entire supercomputer where every single molecule is a tiny processor contributing.
So it's not just a faster processor. It's like having an unfathomably large number of processors all working in concert. That's a completely different way of thinking about computation.
It is a different paradigm. And while this might sound like something straight out of a futuristic movie,
It does a bit.
the field of DNA computing actually has a history that stretches back several decades.
Really? I assume this is a very recent development.
Well, the initial theoretical ideas were actually floated by the legendary physicist Richard Feynman way back in 1959 when he first talked about the potential of nanotechnology.
Feynman, wow.
But the real breakthrough that kickstarted the field came in 1994 with Leonard Adleman's remarkable experiment. He used DNA to solve the Hamiltonian path problem.
Which is a famously difficult problem,
A
right?
notoriously difficult computationally, yes.
And the results were quite astonishing, if I remember correctly.
Indeed. His DNA-based system achieved a processing speed equivalent to around 100 teraflops.
100 teraflops.
Yeah.
In 1994.
Exactly. To give you some context, the fastest supercomputer of that era could only manage about the 36 teraflops.
Wow.
So for that specific, carefully chosen problem, DNA significantly outperformed traditional silicon-based computing. It was a proof of concept that really opened people's eyes.
That's a pretty dramatic comparison. Okay, so fast forward to today,
Yeah.
and we have Dr. Fei Wang's team. What makes their work with these DPGAs such a significant step forward?
Right. So Dr. Wang's team has built upon that foundational work and really taken the concept of DNA computing to an entirely new level of complexity and potential.
Okay.
Their DNA-based system is capable of performing an astounding 100 billion concurrent tasks.
100 billion.
That's the number. It's a truly massive increase in scale, and their publication in Nature really underscores the importance of this achievement.
And the core promise of this work seems to be particularly relevant to our health and well-being, right?
That's a key focus, yes. The team believes this technology has the potential to fundamentally change how we detect and diagnose diseases.
How so?
By being able to support over 100 billion unique circuits, they're essentially creating a platform for incredibly sensitive and specific analysis of biological samples. Imagine being able to detect the earliest signs of disease at a molecular level with unprecedented accuracy.
So the aim isn't just about building faster versions of our current computers. It's about creating a fundamentally different kind of computing system
Exactly.
that can interface directly with biological systems.
Precisely. Their vision is to create computing systems that operate independently of silicon using biological mechanisms to achieve what they call intelligent diagnostics.
Intelligent diagnostics, I like that.
Think about a system that can analyze, say, a blood sample or other bodily fluid and identify disease markers with remarkable precision and speed.
And these DPGAs are central to making this happen. What exactly are they, and how do they achieve this remarkable flexibility?
Okay, DPGAs, as we said, DNA-based programmable gate arrays. They're essentially constructed from short, single-stranded DNA molecules called oligonucleotides.
Oligonucleotides?
Okay, or oligos for short. These short DNA sequences act as the basic building blocks. What makes them so flexible is that you can program them to perform different computational tasks by introducing specific molecular keys.
Molecular keys.
Yeah, think of it like having a set of Lego blocks that can be assembled in countless different ways depending on the instructions you provide. These instructions come in the form of other carefully designed DNA molecules.
And they've actually demonstrated the power of this by tackling some interesting computational problems, haven't they?
They have indeed. In one demonstration, they connected 3 DPGAs using around 500 DNA strands in total to successfully solve quadratic equations.
Quadratic equations with DNA.
Exactly. That's not a typical application you'd associate with biological molecules.
Definitely not.
And in a separate experiment, they designed a DNA circuit that could accurately calculate square roots. These examples really highlight the fundamental computational capabilities of their platform.
But perhaps the most compelling demonstration was in the realm of medical diagnostics, specifically in identifying markers for renal cancer.
Exactly. That was really impressive. They developed a DPGA that could very accurately distinguish between different types of small RNA molecules, including those that are known to be indicators of renal cancer. The way it works is these RNA molecules are introduced to the DNA circuits. If the target RNA is present, it triggers a specific series of chemical reactions within the DPGA.
A cascade.
Right, a cascade ultimately leading to the emission of a fluorescent signal. Light.
So you can see the result.
Precisely. This light signal can then be detected and used to determine whether the cancer-related RNA is present in the sample. This has enormous potential for early and accurate disease detection.
So how are these DPGAs physically constructed, and how do they operate at that tiny molecular level?
Okay, the architecture is based on those short, single-stranded DNA oligos we talked about. A key innovation is the use of generic single-stranded oligos as a consistent signal carrier throughout the system.
Why is that important?
Well, this uniformity helps to minimize unwanted interactions. Think of it as reducing static on a radio signal and ensures the computations are accurate. That's vital for building larger, more complex circuits.
Makes sense.
When the input molecules, like those RNA cancer markers, are introduced, they initiate specific chemical reactions within the DNA circuit. These reactions are carefully designed to cascade through the system, eventually producing output molecules engineered to emit fluorescent light. That allows researchers to read the result of the computation.
It sounds like they've also incorporated some pretty sophisticated design principles into their DPGA prototype, borrowing ideas from traditional computer chip design.
They have indeed. It's quite clever. The prototype includes features like 4 on-chip configuration contexts, which essentially allows it to switch between different program tasks very quickly.
Like multitasking.
Sort of, yeah. It also has DRAM configuration cells, like temporary memory storage within the chip, a non-intrusive background loading mechanism for updating the programs, an automatic refreshing system for maintaining the integrity of the memory elements, and a wide bus architecture, which is like a high-speed data highway designed for quickly loading new configurations onto the DPGA.
So really taking inspiration from silicon.
Absolutely. Features analogous to what you'd find in advanced silicon-based chips, but implemented using DNA.
This marks a significant shift in the field, doesn't it? Moving beyond just solving very narrow specific problems to creating more adaptable and reusable computing platforms.
Absolutely. Early DNA computing experiments often focused on tackling very specific, sometimes quite theoretical computational challenges.
Right, like that Hamiltonian path problem.
Exactly. But the development of these rewritable DPGAs signals a real move towards what the researchers are calling general-purpose DNA computing.
General purpose.
The ability to essentially erase a previous program and load a new one is fundamental. It's analogous to the rewritability we take for granted with our computer hard drives.
Yeah, of course.
And it opens up the potential for DNA computers to tackle a much broader range of problems. This versatility is a critical step towards making the technology commercially viable and applicable in a wider array of real-world scenarios.
Now, one of the inherent challenges with working with molecules floating around in a liquid is the issue of random collisions. How do they ensure that the molecules interact in the precise way they want them to for computation rather than just, bumping into each other randomly?
That's a crucial challenge. You're right. In a liquid environment, you don't have the directed flow of electrons like in a silicon chip. It's more chaotic.
So how do they manage that?
To address this, the team employed a clever technique called spatial compartmentalization. They used intricate DNA origami structures.
DNA origami. That sounds incredibly intricate.
It is. Think of it like microscopic paper folding, but using long strands of DNA folded into precise three-dimensional shapes at the nanoscale.
Wow.
These origami structures create what they call registers. They provide a level of physical organization and directionality, allowing for more controlled and predictable interactions between different parts of the DPGAs.
So the origami structures act like guides or channels.
In a way, yes. These nanoscale structures can compress the three-dimensional output of one part of a circuit onto a two-dimensional rewritable surface within the origami structure.
Okay.
This significantly speeds up the process of transferring signals between different DPGAs that are connected together. They got it down to under an hour in their experiments.
That's a big improvement.
It is. They also developed a special signal amplifier to read the signals stored on the surface. It's a really ingenious way to impose order and directionality on what could otherwise be a very messy molecular environment.
And just to reiterate the key takeaway here, the fact that these DPGAs are reconfigurable is what makes them so powerful for a wide range of potential applications.
Exactly. A single DPGA with 24 addressable dual-rail gates. Think of these gates as the basic logic units can be programmed to implement over 100 billion unique circuits.
100 billion again. That number is staggering.
It really is. This reconfigurability, directly inspired by those FPGAs used in traditional computing, is what makes this technology so promising for tackling a vast array of different computational tasks.
So just to summarize the impressive feats they've demonstrated, these DPGAs can solve mathematical problems, distinguish between disease-related molecules, and even be integrated with conventional electronic systems.
That's right. They've shown their ability to solve quadratic equations, calculate square roots, and perhaps most importantly, accurately differentiate between different types of small RNA molecules associated with renal cancer.
Right.
And the successful integration with an analog to digital converter to classify these disease-related microRNAs really highlights the potential for practical applications in medical diagnostics.
Okay, so we're starting to get a real sense of the exciting potential here. But how does this compare to the supercomputers we already rely on, those based on silicon chips? What are the fundamental advantages of DNA computing, and maybe where does it still face some significant challenges?
That's a crucial question. Let's break down a direct comparison, looking at things like, processing speed, energy efficiency, data storage capacity, and the fundamental computing architecture.
Starting with speed, we saw that Adelman's early experiment showed surprising speed in the 90s. How does that compare to the speed of silicon-based systems today?
Right. Well, while Adelman's work gave us a glimpse of DNA's theoretical potential with that impressive 100 teraflops result,
Yeah.
it's important to understand the different ways these systems operate. DNA's inherent strength lies in its massive parallelism, performing enormous numbers of operations simultaneously.
Right, the trillions of molecules.
Exactly. Traditional silicon, at its core, tends to process information more sequentially, one step after another, although incredibly quickly.
So what are the theoretical numbers?
Theoretically, the potential processing power of DNA, using certain reactions like ligation, is estimated to be in the range of 1014 to maybe 120 operations per second.
That's huge.
It's vastly beyond the capabilities of even the most advanced silicon chips, which operate more in the range of 108 to 1012 operations per second. Some theoretical models even suggest a certain mass of DNA strands could be thousands of times faster than current supercomputers.
But there's a significant difference between that theoretical potential and the speed we see in practical applications, isn't there? I mean, you mentioned an experiment taking 100 hours.
Exactly, that's the catch. While the sheer number of parallel interactions is immense, the individual biochemical reactions themselves are generally much slower than electronic processes in silicon.
Oh, yeah.
That early square root calculation taking over 100 hours is a stark reminder. So it's a bit of a paradox. You have a vast number of things happening at once, but each individual step might take longer.
Plus the setup and readout time, presumably.
Right. You need to consider the time for sequential steps within an algorithm and the practical challenges of getting inputs in and reading results out the I/O overhead.
So it's not just about how fast a single operation is, but the overall time to complete a useful computation.
Precisely. However, researchers are actively working to overcome these speed limitations. We're seeing techniques like localized DNA circuits, fixing strands to a surface, which could potentially cut computation time.
Right.
And as we discussed, those DNA origami registers dramatically cut down signal transfer time between components. There's even been a proof of concept showing all reactions completed in a single tube within about 90 minutes.
Okay, so progress is being made on speed.
Definitely.
Right.
While there's still a gap, progress is happening.
What about energy efficiency? That's a huge consideration for the massive data centers that power our digital world. They consume enormous amounts of power, especially for cooling.
This is an area where DNA computing truly shines. It's estimated that DNA can perform around, get this, 2 by 1019 operations per joule of energy.
Wow. Compared to silicon.
Silicon is roughly 109 operations per joule. So orders and orders of magnitude more efficient.
That's incredible.
An individual DNA reaction consumes an incredibly tiny amount of energy, about 5 by 1020 joules. And crucially, many of these reactions can occur at room temperature.
No massive cooling required.
Exactly. You avoid the enormous energy costs associated with cooling traditional data centers. This inherent energy efficiency makes DNA computing very attractive from a sustainability perspective, green computing.
That's an absolutely staggering difference in energy efficiency. Almost hard to comprehend. Now let's talk about data storage. We hear about DNA having an immense capacity for holding information. Can you give us some real-world comparisons to understand the scale?
The data storage density of DNA is almost mind-boggling. 1 cubic centimeter of DNA could theoretically store more information than 1 trillion CDs.
1 trillion in a cubic centimeter.
That's the theoretical estimate. The data density is around 18 megabits per inch, vastly superior to hard drives.
Okay, give me another one.
Okay, how about this? 1 gram of DNA could potentially store up to 455 billion gigabytes.
Billion.
Gigabytes!
Yeah, that's 455 exabytes,
Yeah.
or 215 petabytes.
Which is equivalent to...
Roughly 500 million 1 terabyte hard drives in 1 gram.
That's insane.
Some projections suggest all the data humanity is expected to generate by, say, 2025 could theoretically be stored in something the size of a ping pong ball.
Wow.
Or in about 1 kilogram of DNA.
It's not just about how much you can store, but also for how long you can store it, right? Stability.
Exactly. DNA is an incredibly stable molecule, especially when stored properly. It can preserve information for tens of thousands, maybe even millions of years.
Without power.
Without requiring any energy input for storage itself. This makes it an ideal candidate for long-term archival storage.
Like libraries or historical records? Precisely. We're already seeing companies like Catalog demonstrating this by encoding things like entire literary works into DNA. It seems like this ability to store vast amounts of data for incredibly long periods could be one of the most immediate and, frankly, commercially viable applications.
So, for tasks that require preserving information for centuries, DNA seems like a clear winner. What about that inherent parallelism we talked about earlier? How does that compare to how silicon-based systems try to achieve parallel processing?
Right. DNA's fundamental ability to explore a vast number of potential solutions simultaneously is a truly unique advantage, especially for certain kinds of problems, like complex optimization problems.
How is it different from GPUs or multi-core CPUs?
It's a fundamentally different approach compared to silicon's typical sequential centralized processing. While silicon has made huge strides in parallel processing with GPUs and multi-core CPUs, it still hasn't quite reached the sheer scale of molecular concurrency that DNA offers. Billions or trillions of molecules working at once.
Right.
And the DTGA technology we've been discussing really highlights this unparalleled potential for parallel processing within DNA computing.
It sounds like the initial vision might have been that DNA would eventually completely replace silicon. But the picture that's emerging now seems more like they'll complement each other.
Yes, I think that's the consensus now. The early ideas of DNA as a direct substitute for silicon are giving way to a more nuanced understanding of their complementary strengths.
So DNA for some things, silicon for others.
Pretty much. DNA excels at those massively parallel, highly energy-efficient operations, particularly suited for specific problems like combinatorial optimization or analyzing complex biological data.
Right.
Silicon, on the other hand, remains the dominant force for high-speed, sequential, general-purpose tasks that need a wide range of input and output capabilities. We know it well. It's mature technology.
And we're seeing hybrid approaches.
Exactly. The development of hybrid DNA computing models that aim to combine the strengths of both by integrating enzymatic reactions with electronic components.
Yeah.
That's a really interesting area, trying to enhance reliability and scalability.
So the future is likely mixed.
I think so. A diverse ecosystem of different computing paradigms, silicon, DNA, maybe quantum, neuromorphic computing, all working together in integrated systems. heterogeneous computing.
That makes a lot of sense. It's about using the best tool for the specific job at hand. So to quickly summarize some of these key differences.
Right. We can think of it this way. DNA offers vastly superior storage density and energy efficiency, plus that inherent massive parallelism for certain complex problems.
Okay.
Silicon currently holds the advantage in raw processing speed for sequential tasks and its versatility for broad general-purpose applications with diverse IR needs.
Okay, so given these really unique strengths of DNA computing, what are some of the most exciting and potentially transformative applications that are starting to appear on the horizon? Where could this really make a difference?
The range of potential applications is truly vast and, frankly, very exciting. One of the most promising areas, as we've already touched on, is in revolutionizing medical diagnostics and paving the way for truly personalized medicine.
And the DPGA technology seems to be really central to this potential, being able to program those circuits for specific tasks.
Absolutely. The potential to fundamentally alter how we detect diseases through intelligent diagnostics, finding those specific disease markers in biological samples, is immense.
Like the renal cancer example.
Exactly. Dr. Wang's team's demonstration of distinguishing between those small RNA molecules is a prime example. Imagine a future of real-time disease monitoring that's seamless, minimally invasive, incredibly efficient, and highly accurate.
Detecting things much earlier.
Much earlier, potentially. And it's not limited to just one marker. DNA computing could enable us to analyze multiple disease indicators simultaneously for much more comprehensive diagnoses.
And even treatment. You mentioned theranostics.
Yes, that's the longer-term vision. The theoretical possibility of creating molecular-scale DNA computers that could both diagnose a condition and then initiate the release of a therapeutic drug right at the site.
Diagnose and treat in one system.
That's the concept. The ultimate vision is autonomous disease identification and targeted therapy delivery at the molecular level. Though, of course, there are huge challenges in making this work safely inside living organisms in vivo.
Right, huge challenges there. Beyond medical diagnostics, what other fields could see a major impact? Drug discovery seems like a natural fit.
That's another area that seems ripe for transformation. DNA computing has the potential to significantly speed up drug discovery and development.
Wow.
By accelerating complex tasks like analyzing how proteins fold or simulating intricate biochemical interactions involved in how a drug might work.
And you mentioned AI earlier. How does that fit in?
Right, the synergy with artificial intelligence is incredibly exciting here. DNA computing's parallel processing capabilities could help overcome some current AI bottlenecks.
Like energy consumption.
Exactly. Especially the energy-intensive training of complex models and the processing of massive biological data sets, which AI needs.
So we could potentially see AI and DNA computing working together to discover new and more effective medicines faster.
Precisely. Think about accelerating the identification of promising drug targets, rapidly screening vast libraries of potential drug candidates, designing entirely new drugs from scratch, de novo design, and improving our predictions of how drugs will interact with targets in the body. It's all part of this growing convergence of AI and synthetic biology, with DNA computing potentially providing a crucial substrate for handling the sheer complexity of biological data.
And could AI itself be further advanced by leveraging the unique capabilities of DNA computing? Not just for biology.
Absolutely. The inherent parallel processing power of DNA could significantly enhance AI generally, processing those massive data sets more efficiently, potentially leading to faster training times for complex AI algorithms.
Are there examples of this already?
Well, researchers at Caltech developed a molecular artificial neural network using DNA.
A neural network made of DNA.
Yes, that could even recognize handwritten numbers. There are also ongoing efforts to develop methods for representing numerical data using DNA concentrations and performing nonlinear functions needed for neural networks using molecular interactions.
Okay.
Experimental work has already shown DNA molecular computation can tackle complex computations, pattern recognition, solve mathematical problems. We're even seeing AI models being used to predict things like DNA methylation patterns, crucial for cell identity and cancer research.
We've already touched on data storage as a key application, that incredible density and longevity. Are there other potential uses for DNA's data handling capabilities, perhaps in security?
Yes, definitely. Beyond just archival storage, the extraordinary capacity and density open up fascinating possibilities in areas like cryptography.
How so?
Well, the sheer number of potential DNA sequences and the parallel computation power could potentially lead to highly secure encryption methods. Think about encryption keys that would be virtually impossible to crack using conventional computers.
Molecular encryption keys.
Potentially. There are also ideas about applications in blockchain technology using DNA to create tamper-resistant, decentralized ledgers.
Interesting.
But as we've said, archival data storage really stands out as the most immediate, perhaps most impactful commercial application right now, especially given the exploding global data storage demands.
The data storage crisis.
Exactly.
And just briefly, are there any other promising areas where DNA computing might make a significant contribution down the line?
Absolutely. We could see it being used for highly sensitive environmental monitoring DNA-based biosensors detecting tiny amounts of pollutants or pathogens.
Okay.
It has significant potential in synthetic biology for precisely engineering living systems for biomanufacturing or advanced genome editing. There's even research exploring its use in advanced mathematics, like verifying safety-critical software.
Right.
And as we discussed, its parallel nature makes it potentially well-suited for tackling those complex combinatorial optimization problems, the traveling salesman problem, knapsack problem, things like that.
It's a truly exciting range of potential applications. But with any technology that has this much promise, there are bound to be some significant hurdles to overcome. What are some of the major technical and practical challenges that researchers are currently grappling with?
You're absolutely right. The potential is enormous, but so are the challenges. There are significant technical, practical, and logistical hurdles that need serious attention for DNA computing to reach its full potential.
Let's start with the technical challenges. You mentioned earlier that accuracy can be an issue. Errors creep in.
Yes, accuracy is absolutely critical, and it's a real challenge. The process of synthesizing DNA, the first step, isn't perfect. Errors can occur: deletions, insertions, substitutions of bases.
Like typos.
Exactly, molecular typos. And the likelihood of these errors tends to increase as the DNA sequence gets longer. Plus, the biochemical reactions themselves aren't always 100% precise, which can introduce errors into the results.
And early error rates were high.
In the early days, yes, unacceptable for most practical applications.
But there's been progress in developing ways to deal with these inherent errors, hasn't there? Error correction.
Definitely. Researchers are actively developing a variety of error correction mechanisms. Some are algorithmic, clever ways to design the computation so errors can be caught and fixed.
Okay.
We're also seeing AI-based methods like DNA Former, helping improve the accuracy of retrieving data from DNA storage. And advanced sequencing technologies like codec are dramatically improving the accuracy of reading DNA sequences.
Which you need to check the results.
Exactly. And researchers are even incorporating error-correcting codes directly into the design of the DNA sequences themselves, like you do in digital communication.
Another challenge you mentioned was the relatively slow speed of the biochemical reactions compared to electronics, and the complexities of input-output, getting data in and out.
That remains a significant area for ongoing R&D. Biochemical reactions are just inherently slower than electron flow in silicon. And getting the precise DNA inputs into the system, then analyzing the outputs, often needing complex, sometimes error-prone techniques like gel electrophoresis, which gives you fuzzy analog results that can be slow and costly.
And concentrations matter too.
Yeah, the need for relatively low DNA concentrations in the mix to ensure proper interactions can also slow down the overall reaction rates.
And then there's the big question of scalability. Can we realistically build DNA computers capable of tackling truly complex, real-world problems that require huge numbers of computational steps?
Scalability is indeed a major hurdle. As the problem complexity increases, the required DNA strand length often increases, which is tough for synthesis and handling.
Okay.
More fundamentally, for certain types of hard computational problems, the amount of DNA needed to explore the vast solution space can grow exponentially.
Like the Hamiltonian path example.
Exactly. Scaling that up to just 200 cities would theoretically require a mass of DNA exceeding the weight of the Earth.
Wow. Okay, so not practical for everything.
Right. So while DNA offers incredible density for storage, that doesn't automatically translate to practical scalability for all types of computational challenges. There are current limits on the number of computational steps per experiment. Scaling this for real-world apps is daunting.
So DNA computing might find its niche.
That seems likely. Most impactful applications might be where the problem space isn't exponentially prohibitive, or where we develop novel algorithms that avoid that exhaustive search. It reinforces the idea of DNA finding its place alongside silicon, not replacing it entirely.
But again, we're seeing innovative approaches being developed to address these limitations and improve scalability. right? It's not a dead end.
Absolutely not. Progress is being made. Those solid-state DNA origami registers help speed up information transfer in larger circuits.
Right.
There's ongoing work on speeding up the reactions, maybe working in very small volumes, or fixing DNA to surfaces which could potentially cut computation time by orders of magnitude.
And AI helping with accuracy and speed.
Yes, AI, like DNA Former, for reading data from storage. And the DPGA design itself aims to reduce the number of molecular components needed, which helps scalability.
And the hybrid systems.
Right, those hybrid DNA electronic systems combining enzymatic reactions and electronics are a promising avenue for enhancing both reliability and the ability to scale up computations.
So it's a continuous process of innovation and problem solving as the field advances. Now, shifting gears to consider the broader implications. A technology with this kind of potential inevitably raises some significant ethical and societal questions that we need to think about proactively. What are some of the key ethical considerations here?
You're absolutely right. As DNA computing evolves, it brings a set of profound ethical and societal considerations that we really need to address thoughtfully and probably sooner rather than later.
One of the first things that jumps out is the issue of genetic privacy and data security, especially if we start storing personal information in DNA. That sounds risky.
Exactly. It's a double-edged sword. On one hand, storing personal data in non-coding regions of DNA could potentially offer unprecedented privacy, portability, resilience. Imagine your medical history encoded in your own DNA. But this immediately raises serious concerns about biohacking, unintended mutations in the stored data, and the risk of worsening existing societal inequalities in access and control.
Misuse of genetic information.
The potential for misuse of highly sensitive genetic information is a very real concern. We already grapple with this with existing DNA databases in law enforcement and research.
Yeah.
And DNA computing adds another complex layer. It forces us to confront questions about the fundamental right to genetic privacy, informed consent, data ownership, and the potential for genetic discrimination or stigmatization.
And the potential for genetic discrimination seems particularly troubling if things go wrong, using that data against people.
It is a significant concern. Without proactive measures and robust safeguards, there's a real risk that DNA computing could inadvertently worsen existing inequities.
Like insurance companies.
That's a classic example. You could imagine scenarios where insurance companies might try to use genetic data from DNA computing to deny coverage or raise premiums based on perceived predispositions. Ensuring equitable access for all populations, especially marginalized groups, is an ethical imperative.
The idea of dual-use capabilities also feels particularly relevant here, given that we're dealing with biological systems. The potential for harm.
Absolutely. The potential for misuse of this technology for harmful purposes, like developing new forms of bioterrorism, is a critical ethical dilemma we must confront head-on.
How do we manage that?
We need extreme caution and careful consideration of how the knowledge and applications are developed and disseminated. Organizations like the World Economic Forum and the Nuclear Threat Initiative are already advocating for things like standardized screening in DNA synthesis to try and mitigate some risks.
What about the environmental impact? While the operational energy efficiency seems like a big plus, what about the large-scale production and eventual disposal of all this synthetic DNA?
That's a very important question that needs more focus. While the energy efficiency during operation is great, we need to consider the entire life cycle.
Cradle to grave.
Exactly. The large-scale production of synthetic DNA, the chemicals involved, and the environmental consequences of its disposal need careful management to prevent unintended ecological disruptions. It's possible the overall environmental footprint, factoring in everything, might not be quite as low as the operational efficiency alone suggests. Responsible development needs to address this.
So it really underscores the need for us to develop comprehensive regulatory frameworks and ethical guidelines right from the outset, not after the fact.
Precisely. These recurring ethical concerns, privacy, discrimination, dual use, environment, they aren't minor issues. They're fundamental challenges needing proactive attention.
And the tech itself is hard to track.
Well, the potential for DNA computers to operate invisibly within cellular environments adds complexity to oversight. There's a clear need for robust regulations and equitable access to the tools, like sequencing and synthesis.
Is regulation keeping up?
Currently, regulations are often ambiguous or inconsistent across different regions, which can hinder responsible innovation. We need clearer frameworks. Ethical guidelines, like those from the Ethics DNA Project, emphasizing values like autonomy, community well-being, reason, experience. They can help guide decisions around trust, consent, ownership.
Hmm.
Ultimately, we need a broad societal conversation about the intended purpose of this powerful technology and embrace a more anticipatory approach to ethics and regulation.
Before problems arise.
Exactly. Failing to address these concerns proactively could lead to public distrust, overly restrictive policies, and unintended harms down the road.
These are certainly a lot of complex issues to navigate. But looking ahead, despite these challenges, the commercial potential for DNA computing seems substantial. What does the future hold in terms of market growth, technological maturation, and when might we start seeing these technologies move out of the lab and into practical use?
The projected growth is quite significant. Current estimates suggest the DNA computing market could grow from around, say, $220 million in 2024 to nearly $1.4 billion by 2030.
That's a big jump.
It's a very healthy compound annual growth rate, over 35%. And this is part of the larger boom in the synthetic biology market overall, projected to hit maybe $70 billion by 2030, especially in healthcare, life sciences, food, agriculture.
And within DNA computing, what's leading the way commercially?
Right now, DNA-based data storage is widely seen as the most promising research area with the clearest path to near-term commercialization, though much is still proof of concept.
But a market is forming.
Definitely. The emerging DNA data storage market alone is projected to reach over $3 billion by 2030. We're seeing a surge in startups focused on DNA storage and related nanotech, increasing competition, driving innovation.
Are the big tech companies involved?
Interestingly, yes. Even established electronics giants are actively exploring hybrid molecular electronic computing architectures. There's growing market recognition of DNA computing's unique value, especially for archival storage and specific parallel tasks.
And in terms of key milestones and realistic timelines, when might we expect to see this technology really mature and become more widely adopted? When does it leave the lab?
Well, we're seeing exciting foundational work now on large-scale, high-speed DNA circuits, progress on tools for visual debugging and automating DNA algorithms.
Okay.
For DNA storage, the big goal is cost reduction, bringing DNA synthesis costs down, potentially reaching a point where it's affordable for long-term archival storage, maybe around $1 per terabyte read-write, perhaps by the end of this decade.
So practical archival storage within 10 years.
Some experts predict exactly that, yes. The
10.
vision of having functional biological computers operating in labs is likely within the next 10 to 20 years.
And related fields.
We're also anticipating continued maturation in areas like cell-free biomanufacturing and a greater convergence of AI, synthetic biology, and automation within the next 5 to 10 years.
So commercialization happens in phases.
Likely, yes. Phased commercialization with data storage and specific diagnostic tools, probably preceding widespread adoption of more general-purpose DNA computing.
And better tools help.
Crucially, continued advancements in DNA synthesis and sequencing technologies from companies like Illumina, Oxford Nanopore are making the computational use of DNA increasingly feasible and cost-effective. That's a key enabler.
So to bring it all together, it sounds like DNA computing is a rapidly evolving field with the genuine potential to spark a computational revolution through its incredible parallelism, data density, and energy efficiency,
Absolutely.
with the development of DPGAs representing a really exciting and significant step forward.
A major milestone, yes. And the interdisciplinary nature of this field, chemistry, biology, engineering coming together, will be crucial for continued progress.
And while we still face some significant technical and ethical challenges that need to be carefully addressed,
Can they really do?
the near-term applications in areas like medical diagnostics, personalized medicine, and especially long-term data storage look incredibly promising, alongside the more distant but equally exciting possibilities in AI and other diverse fields.
And it's important to reiterate that the future of computing will likely involve a complementary relationship between DNA-based systems and traditional silicon-based technologies, leveraging the unique strengths of each. Not necessarily replacement, but augmentation.
Right, using the right tool for the job. And ultimately, our ability to overcome the existing technical hurdles and establish proactive ethical and regulatory frameworks will be essential for ensuring the responsible and widespread adoption of this fascinating and potentially transformative technology.
Indeed. It leaves us with a thought, perhaps, for you, the listener. How might this fundamentally different approach to computation reshape our understanding of information processing, maybe the very nature of technology as we know it? And what broader societal shifts might accompany this increasing convergence of biology and computation? It's a truly remarkable frontier to continue exploring.